Hello and welcome to some biology class. Friends, today we shall discuss about ribosome footprinting. Ribosome footprinting, also known as ribosome profiling, is a technique which can measure ribosomal abundance and the translation at a genome-wide level. This technique is very precise in delineating the translating regions of the genome, thereby revealing the full coding potential of the cell. The technique of ribosome footprinting was developed by Nicholas Ingoglia and Jonathan Weissman. This technique produced a global snapshot of all ribosomes that are active in a cell in response to a stimulus at a particular moment. All these ribosomes which are active in a cell at a particular moment constitute a translator. So the technique of ribosome footprinting provides an idea about all the ribosomes that are active in a cell at a specific moment. This technique also gives us an insight about all those proteins which are getting actively translated at that moment. The technique of ribosome footprinting is based on the fact that the mRNA present within the ribosome can be isolated by the use of ribonuclease, which digest only the mRNA region which are present outside the ribosome. So in this process, the unprotected mRNA region is digested and the mRNA which is present within the ribosome are protected. These protected mRNA region which are present within the ribosome are known as ribosomal protected fragments or ribosomal footprints. The first step in ribosome footprinting is the lysis of cultured cells or the collected tissue sample. The sample could be pre-treated with drugs or external stress conditions in order to investigate as how these external factors impact the cells or tissues translationally. After culturing the cells, the samples are treated with translation inhibitors in order to immobilize the ribosomes on the mRNA. Classical inhibitors such as cycloheximide, anisomycin and emetine, which inhibit the translation elongation, are commonly used for ribosome footprinting. But if one is interested to inhibit the ribosome at the initiation site, then specific inhibitors such as harringtonin and lactibidomycin can be used. In lysis, the sample is broken down either enzymatically or mechanically in order to release the cellular content for further treatment. In the second step, the lysate is treated with the enzyme ribonuclease, which will digest the exposed unprotected mRNA regions which are present outside ribosomes. But the mRNA regions which are present within ribosomes will be protected from digestion. For example, this is a mRNA which is associated with 3 ribosomes. When treated with ribonuclease, the enzyme will digest only these exposed, unprotected mRNA regions. But the mRNAs which are present within ribosomes will be protected from digestion. These protected mRNA regions are called ribosome protected fragments or ribosome footprints. In the next step, the ribosome mRNA complexes are recovered by sucrose gradient centrifugation, or else They can also be retrieved by gel filtration or affinity purification. Similarly, the proteins are also removed. Next step is size selection, in which RNA fragments which are between 26 and 34 nucleotide longs are identified. The ribosome footprint should be between 26 and 34 nucleotide longs. To achieve this, the RNA fragments are separated by polyacrylamide gel electrophoresis. The use of a size selection marker allows us to identify the RNA bands which contain the RNA fragments of specific size. After identifying the RNA band which contain the ribosome footprint, next step is to excise the gel that contain the RPF by using a scalpel. Then the RNA fragments are extracted and purified. Next step is library preparation. For library preparation, first step is RNA depletion. In RNA depletion, the ribosomal RNA, which is the predominant RNA present in the cell, is removed so that we will get better ribosome footprints. After rRNA depletion, the ribosome footprints are reverse transcribed to cDNA by using the enzyme reverse transcriptase. The enzyme reverse transcriptase requires a RNA primer. To overcome this, the ribosome footprints can be tailed by using a polymerase, or else they can be ligated with a RNA linker of known sequence. These ligated mRNA fragments are then reverse transcribed to cDNA. In the next step, the cDNA fragments are amplified by polymerase chain reaction, which amplify the sequences in exponential manner. After PCR amplification, the library can be sequenced to generate a bioinformatic library which can be subjected to different analysis depending upon the aim of the experiment. So friends, this is all about ribosome footprinting. For more updates, please subscribe my YouTube channel, Sam Biology Class.